Myself, Dr. B. Venkata Swamy, Associate Professor, the Chemistry Department, Basic Sciences and Humanities. Course name is Biochemistry. The today's topic is protein structure. The protein structure have the 4 levels of the structure. I am concentrating particularly for the 3D structure of the protein. This is the complete involved in this structure is the 4 structures are available in this one: the primary structure, secondary structure, tertiary structure, and the quaternary structure of the protein. Initially, before starting this protein structure, this is the linear sequence of amino acids linked by the peptide chain. The peptide chain formula is the C double bond O dash NH is the peptide bond forming. One amino acid to another amino acid linking. The dehydration occurs from the protein molecule. One peptide bond occurs. This is the structure of the protein structure. First one, this is the first starting. The first one is the one amino acid. This one amino acid. And the second one is the second amino acid. Dehydration occurs in this one, removal of the water molecule. The forms the protein molecule. This is the peptide bond formula, the C double bond O and NH. This is the peptide bond formula. The joining for the initially formed one peptide bond only. This is a possibility for the many peptide bond formulas. The possibility there, that is combining again another amino acid forms the another amino acid, the removal of the water molecule, combining the protein. See, this is the sequence arrangement of the sequence of the amino acid sequence that forms the protein molecule. This is the primary structure of the protein molecule. The peptide bond, the formula of the peptide bond is this is the peptide bond formula. C double bond N is the combining the structure for the peptide bond. The structure of the protein, this is the polymer of the amino acids made up of the one or more polypeptide chains. The numbers and sequence of amino acids residues in protein different in different proteins. Every protein is a native state. That is a 3-dimensional structure. That is also called the conformational structure of the protein molecule. Protein molecule. That is called the 3-dimensional structure of the protein or the 3D structure of the protein molecule. This is the coming to the first, the primary structure arrangement. The arrangement of the primary structure is the sequence of amino acid. The sequence of amino acid, the first primary structure. The amino acid sequence of the cysteine, the lysine, cysteine, glycine, leucine, valine, alanine, primary structure. This is the secondary structure forming is the folding of the primary structure of the protein. That is called the secondary structure of the protein. Folding. The folding or the twisting. This occurs the alpha helix and the beta, the formations are both are possibility in the secondary structure. Tertiary structure name is called the quaternary. The tertiary structure is the conformational structure is the tertiary structure. Here the alpha and the beta helix are observed in the tertiary structure. The quaternary structure are the 2 alpha helix and the 2 beta, the pleated sheets are present in the quaternary structure of the protein molecule. These are the 4 orders. The 4 levels are the 4 orders of the protein molecule. The first is the primary structure, that is the sequence of amino acids arrangement is the primary structure. The secondary structure is the folding or the twisting of the primary structure, that is called the secondary structure, observed in the alpha helix and the beta pleated sheets are observed in the secondary structure. Tertiary structure is the conformational structure of the alpha helix and the beta helix structure is called the tertiary structure of the protein. This is also called the conformational structure and also called 3-dimensional structure of the protein, 3D structure of the protein. Quaternary structure present in the 4 2 alpha structures and the 2 beta structures are present in the quaternary structure of the protein molecule. This is the primary structure. Initial, the one is the primary structure, arrangement of the sequence of the primary structure. The formula for the C double bond O NH, the one amino acid. The second peptide bond, this one. The third peptide bond, this one. Fourth peptide bond, this one. And the fifth peptide bond, this one. And this arrangement of the peptide bond, the formation, the sequence of amino acids called the primary structure. So coming to the secondary structure contain the folding or the twisting of the primary the structure of the protein. The forms, the 2 forms. One is the alpha helix form, another one is the beta pleated sheet form. This is the beta pleated sheet form, this one. Beta pleated sheet form, this one. This is the alpha helix structure of this one. Coming to the third tertiary structure is the 2 alpha and the 1 alpha and 1 beta conformational arrangement of the protein structure is called tertiary structure of the protein. The coming to the quaternary structure is the 2 alpha. This is 1 alpha. This is another alpha. This is the 1 beta. This is another beta. 2 alpha and 2 beta structures are present in the quaternary structure of the protein molecule. The protein structure, the peptide bond is initially the conformation structure, the required for this one. The 4 levels are the 4 orders of the primary structure. First one, this is the linear sequence of the amino acids joined by linking called the peptide chain. Secondary structure is the alpha and beta pleated sheets arrangement is the secondary structure. This is the primary structure of the protein. This is the not chemical formula. This is the physical structure of the protein molecule. The protein molecule physical. Each round circular is the one amino acid. Between joining link is called the peptide bond. The peptide bond C double bond O NH is the peptide bond formula. This is the physical structure of the protein molecule. Coming to the secondary structure, I am taking the example for the primary structure and the secondary structure purpose. Human insulin taken for this one. The alpha chain and the beta chain forming the structure is called the primary structure. Insulin have the linking between the primary chain and the secondary chain. Here the between link is called the sulfide bonds. Disulfide bonds are present in the joining link, primary and the secondary. This is alpha helix. The structure of the alpha helix is the required for the 3.6 amino acids required for per turn of the helix. Right, the covered distance of this one is the 0.4 nanometers of the distance for the each amino acid sequence. Each residue represents 0.15 nanometers advance along helix axis. The side chain position outside of the cylindrical. This is the cylindrical, the shape of this one. This one amino acid to amino acid. This is the one turn required for the 3.6 amino acids are required for the one turn of the helix. That is the alpha helix structure. Coming to the beta pleated sheet. This is the detailed structure of the secondary structure. This one also same cylindrical shape. 1, 2, 3, 4, 5, the 6, 7, 8, 9 amino acids arranged in this one. The distance for the 1 to 5. 1.1 to 5 angstroms in the rise, 100 angstroms in the rotation. This is the cylindrical shape structure is the secondary structure of the protein. This is alpha helix diagram for the human insulin purpose. Take an example. Coming to the protein, the beta pleated sheets also arrangement of the hydrogen bonds between the peptide bonds arrangement is present in the beta pleated sheets, and disulfide bonds also present in the each hydrogen bonds and to one amino acid to another amino acid, or hydrogen to hydrogen, right? The carbon to carbon, nitrogen to nitrogen. Between the dots are the hydrogen bonds. These are the protein structure. This is the two types of beta pleated sheets are present. The direction for this one is the parallel beta pleated sheets also possible. Parallel only moves the one side of the direction, only moving the component called the parallel beta pleated sheet. The second one is the two anti-parallel direction. Observe, this is the anti-parallel. This is the one direction. This is the another direction of the protein, the amino acid sequence move. The beta pleated sheet contain the parallel possibility availability there, and the anti-parallel also availability in the human insulin beta pleated sheets. Next, this is the complete structure of this is the anti-parallel direction. This is the anti-parallel direction. This one, this is the direction is the anti-parallel. This is the one direction. This is the another direction. This is the another direction. The second direction. This is called the anti-parallel direction. Parallel, coming to the observe, this structure is only the one-way direction. This also same direction only. This type of the arrangement, the movement is called the parallel beta pleated sheet direction. This is the protein structure for this one, the alpha helix cylindrical shape, the folded structure. This is like the rod like shape structure is called the one helix, or the helix structure is a folded structure. This one is the beta pleated sheets is the anti-parallel. This one direction for parallel, another one is the anti-parallel. The two possibilities are present in this one. Hydrogen bonds are perpendicular to a polypeptide chain. Actual distance for this one amino acid between the amino acid to amino acid is the 3.5 angstrom diameter. The protein structure, the tertiary structure. This is the very important for the tertiary structure, also called the three-dimensional structure of the protein. This is the spatial arrangement of the amino acid sequences are called a three-dimensional structure or the 3D structure of the protein. This is also called the conformational structure of the protein. Here arranging the alpha helix sheet and the beta pleated sheets are arranged in the conformational arrangement. The spatial arrangement of the amino acid sequences is called the tertiary structure of the protein. Here taken the example for the myoglobin. The tertiary structure example, the purpose explaining purpose, taking the example for the myoglobin for the tertiary structure. Right? Is the advantage for the tertiary structure comparatively to the other structures are this: once folded into the structure 3D, biologically this is the active or the conformation is maintained by hydrophobic interactions, hydrophobic interactions and hydrogen bonding, and the electrostatic forces including the salt bridges, van der Waals interactions and covalent to disulfide bond. These are the advantages for the 3D structure or the tertiary structure of the protein. This is called the conformational structure of the protein. Now coming, this is observe this one is the open loop structure alpha. This is the alpha helix structure. The open loop form. This is the beta pleated arrangement of this is the beta pleated sheets. This is the helix structure is called the helix structure. Folded structure is called the alpha helix. This is the complete structure for this one is the tertiary structure is this is a connecting link for one amino acid and the disulfide bond. This is the beta pleated sheet. This is state arrangement. The folding structure is called the alpha helix structure of the protein molecule. Coming to the fourth structure is called the quaternary structure. One polypeptide chair. Here the taking the example for the hemoglobin, iron, the hemoglobin content. Here arrangement of the two alpha helix structures and two beta pleated sheets are present in the structure. The linking between the polypeptide chains arrangement. These interactions may be covalent link for the non-covalent interactions of the protein molecule. This is the protein molecule for the quaternary structure of the protein. Example for the hemoglobin. The hemoglobin example. This is the one alpha, the first alpha 1. The alpha helix alpha 1 and this is the alpha 2, the second one, the beta pleated sheet 1, beta pleated sheet 2. This is the quaternary structure of the protein. This is the iron containing hemi group containing the hemoglobin. Hemoglobin taking example for the iron. And this is complete the protein structure. Hemoglobin molecule contain the iron, alpha chain and the beta chain. Alpha chain this indicating the red color and beta chain, beta pleated sheet, beta chain. And this complete chain, the alpha 1 chain, alpha 2, the beta 1 and the beta 2. Two alphas and two beta sheets are present in the quaternary structure of the protein molecule. These are the general examples I have taken for the explanation of the complete process. Initially taken for the amino acids. The one brick is called the one amino acid. Right, the primary structure arranging the one brick is the one amino acid, second one is second amino acid. Between linking, this link is called the peptide bond linking. The arrangement of the structure, the wall arrangement for this one is the primary structure. The secondary structure here, the bends are the twisted. Observe this one, this bending. The bending or the twist in the secondary structure of the protein. Coming to the tertiary structure, self-contained the rooms. This is the one, right? This is the one. This is the third one, right? The self-contained rooms is the tertiary structure. Conformational arrangement in the alpha and the beta pleated sheets. That is called the tertiary structure or the 3D structure or the conformation structure of the protein. Coming to the fourth one, the quaternary structure building with the similar dissimilar rooms. 1, 2, 3. The quaternary structure, two alphas and the two beta pleated sheets arranged in this one, the bricks type of arrangement and the rooms type of or the house type of arrangement is called the quaternary structure of. This is a general example for the concept of the protein structure example. Right? This one is the protein structure complete. The detailed structure for this one is the levels. This is the primary. The first one is the primary structure. The primary structure is the physical structure and the secondary structure, third is tertiary structure and the quaternary structure. The tertiary structure is also called the 3D structure of the protein, 3D structure of the protein molecule. Fourth one is the quaternary structure contain the 2 alpha and 2 beta pleated sheets. Right? This is the complete, the protein, the structure contain the 4 levels orders, the completed. Right? Thank you.
Like, share and subscribe. Hit the bell icon for more updates.